everyone. My name is Tran. I'm a second year Foothill College student, majoring in computer science, and I've been working at Stand Biotools in their research and development department on their X9 high throughput genomic system. So to understand what the X9 does, we have to know what real-time PCR is, and it's a laboratory technique that used to amplify and quantify specific DNA or RNA sequences. And the X9 basically automates and simplifies that process. And how it does that is it works in conjunction with the company's microfluidics technology chip to condense the workflow into 4 simple steps. So you start with preparing the master mixes, samples, and detection assays. Then that gets pipetted into their dynamic array, which then goes into the X9 machine to load and go through the thermal cycling, and then data and images is captured throughout the cycles, and all of that will be available for analysis at the end. So why do we care? Well, these tools have a broad range of applications from human health, fisheries, animal breeding, and sustainable agriculture research. In the realm of human health, one branch of science that these tools could help accelerate is called pharmacogenomics. They basically aim to study how an individual genome dictates their drug response. And inadvertently, by supporting pharmacogenomics research, it also helped aid in fields like psychiatry, why is one drug better than the other, and what drug combination work best together in the sense of addiction research, how drug affects the brain reward function, and in pain management, what's the optimal dosage and response when it comes to certain drugs. In fisheries and animal breeding and sustainable agriculture research, these tools could give researchers deep insights into topics such as molecular assisted breeding, pathogen detection, quality control assessment, all of which are really crucial in sustaining our food sources as well as ensuring their quality and safety. So my focus over the last 10 weeks has been to develop a diagnostic script for their pressure relief valve. What the pressure relief valve is this little component right here on the regulator block. And what the regulator block does is it's the pneumatic components of the instrument. So basically, when the chip get loaded into the X9, what drives the liquid to move within the circuit is pressure. They use compressed air to drive the liquid and thus facilitating the reactions that need to take place. And that needs to take place and observed. So they were having an issue in the field with the pressure relief valve releasing air way too early. And the pressure relief valve is it works similarly to a safety valve on a pressure cooker. So it's a fail-safe component that meant to vent system over pressure, but they were having issue with it just not meeting the spec that they needed to. Thus, their customers were receiving errors when they were carrying out the real-time PCR process. So this is the platform that I have been coding and testing on. Is their proprietary software it's called EKS, Engineering Control System. It's meant for their engineer to go in to write, test, and implement scripts that pertains to the maintenance of the system. So here's the method. I had to start off learning Python. Then I had to familiarize myself with the existing engineering scripts, focusing on the pneumatics aspects of it. Then, of course, I had to familiarize myself with the platform that I would be coding on, do the actual coding and testing. So the logic of the code is basically really simple. Just imagine you have a perfectly fine glass of water, and you know that it takes 5 seconds to fill it up. Basically, what the script does is if there's a crack in that cup, it would detect, because if you're pouring for 5 seconds and it's not meeting a certain level. So here's a little quick snippet of the script. This is running. it put out a fail message in a message box. This is what the field service engineer would see. And everything you see in the console here also get logged into a text output for the engineers to look at later. And of course, after testing, I did team review to get feedbacks on what's optimal, what could be changed, what could be better. And usually that cycle ends with me making the revisions needed to make the script better. Here are some sample results on 5 instruments. And on the top level, it's just, this data is basically just saying, yes, like the script works and this is why it works, because the parameters that we need to meet, it's meeting it. So this is where I left off with my team last week. We said that yes, the script is reliable as a diagnostic test. It can conclusively identify issues, but it's not fully prognostic, meaning that just because an instrument passes the test today doesn't mean that it can't fail down the line. I recommend that you do more testing on other instruments. Of course, more data never hurts. And the next steps is we're releasing the scripts, we're implementing it as part of all the on-site service visits. They're going to update the field service bulletin documentation with it, which is just basically an internal technical documentation repository. And then they will train their field service engineer and product service engineer to run the scripts. I don't know if you guys were able to catch that, but it says anything that can go wrong will go wrong, Murphy's Law. And so I chose that quote to encompass my experience over the last 10 weeks, not out of pessimism, but more so to serve as a reminder to be meticulous with my work, test my code, consider my edge cases, consult with other people who know more than I do, and keep proper documentation of my process. Not to eliminate what can go wrong, but to limit it as much as possible. And of course, acknowledgements. Thank you to my mentors, Bong and Frank, and the rest of my team, Warren Lynette, for being so gracious and generous with their time and helping me navigate this process over the last 10 weeks. Thank you to Foothill College and the Science Learning Institute for funding this opportunity. And of course, Sophia and Marissa also for helping us navigate this very rewarding, but very intimidating process, and just instilling confidence in us when we're just too scared to.